All right. So the first week of embryonic development, we are talking about how is the child going to form from the first day to the seventh day. That's the first week. So the process of progressing from a remember you started as what has to happen is that there has been intercourse, and so fertilization has happened. The female oocyte has merged with the male spermatozoa, and that results into a zygote. And these cells which forms is going to start going through different stages to develop. Now the first 8 weeks of human development is known as a embryogenesis, the period of embryogenesis or organogenesis, because you are basically forming organs for the first 8 weeks. 8 weeks meaning this is like 2 months is the period for forming organs. The period from that point after 8 weeks until birth, that is known as a fetal period, because you've already formed all the organs and then the child is just developing. This seems very easy, but I've seen a question which they've said the fetal period is, or the period of organogenesis runs from 2. So just know that. Now, in this period when you are forming the child, a lot of things, when the child is forming, you're not forming. Okay. A lot of things can go wrong. So the study of embryological origins and causes for defects at birth is known as teratology. So every time you're talking about embryology, you need to know what can go wrong. Why is it that some people are born like this? So when you are talking about the gestation period or the pregnancy period, first week of development is going to begin on ovulation. That day when a woman does remove the egg. Remember, you have got menstrual cycle. Menstrual cycle roughly is for 28 days average. The first day is the day when the woman has blood. 14 days from that day will be the day of ovulation when the egg is released. If that egg is fertilized, then ovulation, fertilization is going, the child is going to start forming. So the first day is counted as that day that the woman released the egg. So this is the day that you start counting from, even to predict when the child is more likely to be born. The last normal menstrual period is going to be counted as the beginning of gestation clinical period. Now the last normal menstrual period. So when last did you have your period? That is going to be counted as the beginning of gestation period or the beginning of pregnancy. And in obstetrics, the study of this is a study of birth, children. It's a branch of science that is going to study that. So the gestation is going to be divided into three trimesters. You are going to have the first 12 weeks, that is the first trimester. And then from the 13th week to the 24th week, that is going to be the second trimester. From the 25th week to the 42nd week, plus 6. We mean plus 6 because if the 42nd week starts, it needs to end. Okay, like to the last day of the 42nd week. That is going to be the third trimester. A normal gestation period should be between 37 weeks to 42 weeks. Any birth that is going to be before 28 weeks, that is known as a miscarriage or an abortion before 28 weeks. The World Health Organization has standardized the fetal viability age. Like if a child is born in this period, then they are okay, they can actually get to survive. So for the World Health Organization, the fetal viability age is 28 weeks. But in Zambia, it is 28 weeks. So if any child is born before 28 weeks, we can barely support them looking at the instruments that we have. So that is a miscarriage or it's an abortion. You can also have premature babies. Premature babies are those babies who are born before the fetal viability age. According to the World Health Organization, that's before 24 weeks. According to Zambia, that is 28 weeks. Now, a premature baby is born, okay, before 24 weeks for Zambia, or sorry, for this was supposed to be for World Health Organization, and 28 for Zambia. Now this word before I'd put here is actually wrong. So a premature baby is a baby born after 24 weeks for WHO, or 28 weeks for Zambia, but less than 37 weeks. Remember, normal is 37 to 42. So, and the viability age is 24 for WHO, and then 28 for Zambia. So what does that mean? So for Zambia, if you are born between 28 and 37, this period, of course you are viable, you can survive, but then you are not in the normal period. So that is known as mature baby. Just take note of that, right? Important. So if a child is born and the child is born with weight below 2.5 kg, the normal weight that a child should be born with is between 2.5 and 3.5 kg. The child is born with a weight below 2.5, that is low birth weight. The child is born with a weight greater than 3.5, that is high birth weight. So the child is microsomic. Say microsomia. The child is too fat. Okay. So normally children who have got who are microsomic, who are big macrosomic, are going to come from parents who are diabetic, parents who have got sugar, have got a high chances of giving birth to children who are macrosomic. Okay. And these children can actually, coming from diabetic parents, have got a high chance of intrauterine death. Just in the womb, the child dies, or after birth, the child dies. And the baby is going to have high levels of insulin, and that is going to cause hypoglycemia. So what happens is this. Remember, the mother is diabetic, meaning the mother has got a lot of sugar. So what is going to happen is that the mother will start to produce insulin, because insulin will cause this sugar to be stored. Now this insulin can penetrate the placenta and it will go to the child, but the child doesn't have a lot of sugar. So the little sugar that the child has will be stored. When you store sugar, it means you are going to have little sugar in blood. That is known as hypoglycemia. So the problem with the mother was hyperglycemia. But then the problem with the child is hypoglycemia because of the insulin that has moved. Okay. So we have talked about low birth weight. So between 1.5 kg to 2.5 kg, that is low birth weight. But between 1 to 1.5 kg, that is very low birth weight. And then between 0.5 and 1 kg, that is extremely low birth weight. So that's how you can classify the birth weight of a child. One thing we need to understand when we're talking about the birth is the female reproductive system. You can see here is the ovary with ovums or the eggs in different levels of the cell, in different levels of development. Got primordial germ cells, the primary oocytes, the secondary oocytes, and also the mature ones, the Graafian follicles. The beginning of the fallopian tube is very wide and is called the infundibulum. You can see it has got these parts which look like hairs, okay, which we call fimbriae. And then as you go away from the infundibulum, you're going to have this long path is also a bit wide. That's known as the ampulla. And then you're also going to have the isthmus and then the intramural, which is the point that you enter the uterus. You can see the uterus also has got different layers. The innermost layer is the endometrium, also of the muscle layer, which is the myometrium, and then the outermost layer is going to be the perimetrium, which is going to be continuous with the coverings or the folds of the peritoneum. We'll understand that later on. So what happens is that on the 14th day in the menstrual cycle, luteinizing hormone is released, and that luteinizing hormone is going to cause ovulation, the release of the egg. And you can see the uterus continues to proliferate. It is continuously becoming big because it is preparing in case the egg is fertilized, it has to be implanted. So that is why it continuously becomes big. What are some physical signs that someone has ovulated? They have removed the egg. There's going to be a rise in basal body temperature. So the body temperature is going to be high, and this is actually used as a contraceptive method. You have a girlfriend or you have got a wife, and you notice that there is a high basal body temperature. You know that ovulation has happened, so you avoid sex that period because pregnancy can happen. And then also there's going to be higher levels of luteinizing hormone. Remember, the luteinizing hormone is going to be released on the 14th day. So if you notice high levels of luteinizing hormone in blood, you know that ovulation has happened. The breasts are also going to become tender, okay, with light spottings and a slight pain or cramping in the side. There is also what is known as the day 21 serum progesterone. Look at what happens here. You can see that progesterone is not really high on the 14th day, but after the 14th day, progesterone is high because progesterone is preparing for implantation. So the egg has been released in case fertilization happened. So progesterone by the 21st day is very high in preparation for the implantation because it is the one which maintains the uterus. And then there's also what is known as a spinner bucket. And this is basically the stretchy quality of the cervical mucus. So if you see the mucus from the cervix, it's going to be that bit of stretchy. So be able to know that the ovulation is happened or it's around the corner. We've also talked about the basal body temperature. Okay, so take note of this. Now fertilization. Fertilization is most going to happen in the ampullary region. The ampulla, this region here, it is very long and it is also not that thin. That is the region where fertilization is more likely to happen. It's the widest part of the tube, and it is close to the spermatozoa. Is actually going to be, it's close to where the egg is coming from, not really far. Now, spermatozoa after sexual intercourse is released in the female reproductive system. It is going to be moving and it can survive like for 3 days in the female. Of all the sperms which are released in each ejaculate, you have got like 20 million sperms, and a normal ejaculate is about 2 to 6 mils. So normally you are going to have like over 50 million sperms. So ejaculating less than 10 million sperms, then that male is infertile. 10 million. Of all those sperms which have been released, only 1% which have been deposited in the vagina is going to enter the cervix. And then they are going to survive there for several hours. Movement of the sperm from the cervix to the uterus is going to occur by the contraction of the uterus and the uterine tubes, and very little by their own movement or their own propulsion. So the trip from the cervix to the oviduct or the fallopian tube takes like 30 minutes or even very slow up to 6 days. After reaching the isthmus, which is the narrow part, the sperms are now going to become less motile and they will cease their migration in the isthmus. And then at ovulation, sperm again will become motile because sometimes the sperms can be released days before ovulation. So they will gain motility after ovulation because the egg that has been released starts to attract the sperms. So it has got chemoattractants. Because remember, the egg has got the cells which surround it, which we call the cumulus ophorus cells. Those have got chemoattractant nature that will attract the sperms. And then they'll be able to swim to the ampulla where fertilization usually happens. This doesn't mean it doesn't happen anywhere else. Is this okay up to here? Are we good? All right. Now, when the sperms reach to where the egg is in the female reproductive system, and then they penetrate the egg has got different layers. All right, it has got different layers, so all those layers have to be penetrated, and not all sperms are going to penetrate those layers, only one. So two things happen when the egg comes in contact with the sperm: capacitation and what we call acrosomal reaction. Remember, we described this in the previous class to say when a sperm is formed in gametogenesis, it is going to be decapacitated. In other words, you cover the head of the sperm so that it doesn't more like start looking for the egg in the male reproductive system. So you need to give it capacity to move. That is known as capacitation. You need to remove that glycoprotein covering, and then it will also undergo the acrosomal reaction. Capacitation is a period of conditioning and it happens in the female reproductive tract, and it lasts for how long? 7 hours. Make sure that you are taking also note of the times and the numbers. So speeding to the ampulla is not an advantage of the sperm. Even if the sperm reaches first to the ampulla, that doesn't mean it is the one that is going to fertilize the egg, because it's only the sperm that has been capacitated that is capable of fertilizing the egg. And much of the or conditioning during capacitation will happen in the uterine tubes and involves the epithelial interaction between the sperm and the mucosal surface of the tube. So the sperm is actually interacting with the mucosa or the tube, the fallopian tube, and that is also disqualifying some sperms which are not supposed to reach the egg. During this period, a glycoprotein coat and seminoplasma proteins are removed from the plasma membrane. Because when the sperm is actually moving from wherever it is producing the seminiferous tubules, as this moves in the reproductive system, it is going to mix with a lot of fluids like the seminal fluids, prostate fluids. So all these and the glycoproteins will cover it and they have to be removed. Only capacitated sperm can pass through the corona cells and undergo the acrosomal reaction. So when you look at the egg, it has got the corona radiator, the cells which cover the egg, the outermost cells. So that layer, the corona radiator, has to be passed. Then it has got the inner layer, which we call the zona pellucida. So two layers have to be passed, the corona radiator and the zona pellucida. Only capacitated sperms can pass through the corona radiator. And then when you pass through the corona radiator, then the zona reaction is going to the acrosomal reaction is going to happen. Acrosomal reaction involves the binding of the sperm to the zona pellucida, and this one is induced by what we call zona proteins, and you have got like zona protein 1, zona protein 2, zona protein 3, those. And this reaction will culminate in the release of the enzymes needed to penetrate the zona pellucida, because first the sperm is interacting with the walls of the zona pellucida. And then remember, the sperm, as at the end of the sperm there is what we call the acrosome, which has got hydrolytic enzymes. So those are going to be released. And what really is released? Acrosines. and trypsins. Take note, acrosins and trypsins are released, and these will be able to digest or to break down the zona pellucida, and then the sperm will be able to penetrate. Okay, and so the phases here will include the phases that happens when the sperm has reached the egg, penetration of the corona radiator, and then penetration of the zona pellucida, and then the sperm will be able to fuse with the oocyte after it has penetrated. So you can see phase 1 is the penetration of the zona and the corona radiator barrier, and then phase 2 will be the entry through the zona pellucida. When the zona pellucida is penetrated, then what will happen is that the spermatozoon, that is the sperm, will then merge with the oocyte. Okay, it will merge with the oocyte. So in phase 1, the penetration of the corona radiator, 200 to 300 million spermatozoa are normally deposited in the female, but only 300 to 500 will reach the site of fertilization, because we said it's like 1% reaches the site of fertilization. Okay, and only one of these sperms will be able to fertilize the egg. It is thought that others are going to just help, more like you're only pushing one, right? This is the one who's supposed to win, so we are all escorting this one. Capacitated sperm will be able to pass freely through the corona radiator, and then the phase 2 will be now penetration of the zona pellucida. So what will happen is that the zona is a glycoprotein shell around the egg, which maintains sperm binding and induces the acrosomal reaction. We'll talk about it. So both the binding and the acrosomal reaction are going to be mediated by ligand ZP3, a zona protein. So please take note, ZP3, okay, will be able to mediate this, but you are also having other zona proteins, ZP1, ZP2. And what will happen is that there's going to be release of acrosomal enzyme. We talked about acrosins and trypsins, okay, which allow to penetrate the zona, and then the sperm will come in contact with the egg. Permeability of the zona pellucida is going to change when the head of the sperm comes in contact with the oocyte. Immediately the head of the sperm enters the egg, and then it comes in contact with the oocyte. Then the zona pellucida becomes impermeable. Other sperm shouldn't enter, because if they do enter, it is going to lead to polyspermy. You end up having multiple pregnancies, which can lead to twins, triplets, and such. Okay, so what is going to happen is that there are going to be enzymes which will prevent the penetration of other sperms to be entering. Only one that has entered should enter. Now phase 3 will involve now the fusion of the oocyte and the sperm. The initial adhesion of the sperm to the oocyte is mediated in part by the interaction of integrins. So the sperm is interacting with the oocyte. What is going to make sure that that happens is the integrins on the oocyte, and their ligands, the disintegrins on the sperm. Okay, just take note, where are integrins found? On the oocyte. The disintegrins, these are on the sperm. And what is this doing? It is aiding in initial adhesion, or the binding of the sperm to the oocyte. After this bind, the plasma membrane of the sperm and the egg will then fuse, and it will become one. Okay, that is because the plasma membrane covering of the acrosomal head cap is going to disappear during the acrosomal reaction, and the actual fusion is going to be accomplished between the oocyte membrane and the membrane that covers the posterior region of the sperm head, the back of the head. So in human take note that the entire head and the tail of the sperm is going to enter into the cytoplasm of the oocyte, but the plasma membrane is going to remain on the surface of the oocyte. The head and the tail enters, the plasma membrane remains outside. You can see the sperm with the head has entered. And so inside the oocyte, you can see we have got the what we call the female pronucleus, and we're also going to have what we call the male pronucleus. Now other reactions will happen. Remember we said that the egg that is released during ovulation is actually arrested in meiosis 2, metaphase 2. And the only way meiosis 2 for the egg can finish is if the egg is fertilized. So as soon as the spermatozoon has entered the secondary oocyte, the egg then is going to respond in three ways. One, there's going to be the cortical and the zonal reaction being activated, the resumption of the second meiotic division, and then a metabolic activation of the egg. How about the cortical and zonal reaction? What happens? So as a result of the release of the cortical oocyte granules, which contain lysosomal enzyme, because when the sperm penetrates the oocyte, there's going to be the release of this which we call the cortical oocyte granules, and they contain lysosomal enzymes. So the oocyte membrane becomes impenetrable to other spermatozoa. The zona pellucida alters its structure and composition to prevent other sperms from entering, and that prevents polyspermy. The zona pellucida is actually going to disappear on the fourth day of fertilization. Very important question. When does the zona pellucida disappear? On the fourth day. And then there's now going to be the resumption of the second meiotic division, because what is going to happen is that the secondary oocyte is going to now finish its second meiotic division because it was arrested in meiosis 2, specifically metaphase 2. Okay. One of the daughter cells which receives hardly any cytoplasm, known as the second polar body, is gone. Now, that's why we started with gametogenesis, because in gametogenesis, we had the primary oocyte forming a secondary oocyte and also a second polar body. Second polar body just goes like that, but the secondary oocyte is the one that will become the definite oocyte, which is fertilized now. Okay, and this definite oocyte, it is oocyte, so it has got 22 it has got 22 chromosomes, which are just body chromosomes, and then one X chromosome. In a sperm, you have got 22 somatic and then one Y, because Y is for males. So a sperm has got the Y. So this 22 plus X is known as the female pronucleus, which will merge with the male pronucleus, which can be 22 plus Y or 22 plus X, because a male sperm can either have the X or Y. That will determine whether you're going to give birth to a male or a female. Up to this point, are we good? Okay. Remember, we have got 23 pairs of chromosomes in our body. 22 of the chromosomes don't determine our sex, only one determines our sex. So 22 plus X means 22, these are other chromosomes, but the sex chromosome is X. It is remember, this is female. If it is in a male, it is 22 other chromosomes plus a Y chromosome, because it's male. Does that make sense? Okay. Another thing that happens when fertilization has happened is there's going to be metabolic activation of the egg. Very, very important. So what happens? Activating factor is going to be the spermatozoon. The sperm is actually what activates the egg. So after fusion, activation may be considered to encompass the initial cellular and molecular events associated with early embryogenesis. Immediately the female pronucleus and the male pronucleus merge, then the child has started forming. You can see the female and the male pronucleus start to merge, and then meiosis 2 is going to finish, and then the first division is going to happen. You can see a division is happening. So you are going to form two cells, okay, two different cells, and this is known as a two-cell stage. Two-cell stage, and life has started. Okay, that is how life starts. Now remember, the female pronucleus supposed to contain 23 chromosomes, haploid. Same with the male pronucleus, contains 23 chromosomes, that's also haploid. Together they'll form 46 chromosomes as a zygote. Morphologically, in other words, in structure, you can differentiate that this is a female pronucleus, this is a male pronucleus. It is all the same. It looks the same. Okay. So during the growth of the male and female pronucleus, each pronucleus must replicate its DNA. It is going to replicate its DNA. If it does not, each of the two cell zygotes has only half of the normal amount of DNA. That's why it has to replicate the DNA. And immediately after the DNA synthesis, chromosomes are going to organize in spindle fibers in preparation for normal mitotic division. Mitosis has to happen. After meiosis, you have got the 23 maternal and the 23 paternal chromosomes splitting, and then you are having a zygote forming with diploid number of chromosomes. So as the sister chromatids move to opposite poles, because you're having one, the chromatid, you have got a chromosome, it looks like an X. So when it is separated on the centromere, those are called sister chromatids. So they'll start to move on the opposite poles in the anaphase, right? And that is what is going to result. This is what you can see anaphase happening. So they've been pulled away like that. And then division can happen. Are you able to notice that remember we said that the primary oocyte forms the secondary oocyte and the second polar body. The second polar body is actually embedded in the secondary oocyte. It's not alone. You can see the polar body here. Just take note of that. Anatomy is a bit interesting the way they formulate questions. Okay. So what will be the result after fertilization has happened? One is that is I saw one exam question which they said list, for that one they said at least five results of fertilization. So one thing is that after fertilization, there's going to be restoration of the diploid number of chromosomes. Remember, it was haploid. The male 23, the female 23. So after fertilization, it will come back to 46. There's also going to be the determination of the sex of the new individual. So if the sperm that is coming carries the X chromosome, then you are going to be female. If it carries the Y chromosome, then it's going to be male, because the egg always contains the X chromosome. And then the result of fertilization, one important one is there is going to be initiation of cleavage. Okay. So without fertilization, the oocyte will be able to die just within 24 hours. But after fertilization has happened, cleavage happens. What is cleavage? Cleavage is where the the female pronucleus and the male pronucleus come together, and then there's this division, and then you have formed two cells. That is known as a two-cell stage. If you don't want to be pregnant, you can use contraceptives. You have got barriers like condoms. You can see each method has got a failure rate. You can use diaphragm, spermicides, which will kill sperms, cervical cap, sponge. You can also use hormonal methods like a combination of estrogen and progesterone, okay, which can be oral, patches. You can also use progesterone only. This one can be injected, or it can be oral or subdermal via the skin. You also have the intrauterine device being used, images contraceptive pills like the morning after pills and those others, which will be made up of progesterone. Okay. Also natural family planning based on symptom. If you just feel that the body is hot, you know ovulation is happening. Calendar based breastfeeding, withdrawal method, coitus interruptus. Though it's a very hard method, it's unreliable because normally you fail to withdraw. Also sterilization, you can as well just cut off the male reproductive system so that sperms are not transported. So all this can happen. They can as well be infertility. Normal ejaculate is 3 to 4 mils. In some others you see literature 2 to 6 mils. And each mil contains 100 million sperms. So normally how many sperms are being released? Like 300 million to 400 million. So any male that releases less than 100 million, 10 million sperms, that one is infertile. Okay. And infertility in a woman can be due to a lot of things. For example, the oviduct can have some infection. The mucus itself in the cervix can prevent sperms from passing. And also immunity to spermatozoa. Every time sperms come into the female reproductive system, you are immune to them. They are destroyed. Or there is no ovulation, that is anovulation, and a lot other things that can cause that. But there are other methods that you can use in case someone can't give birth, like the in vitro fertilization. So you get a human ova, okay, and then, or even an embryo. Now you get to transfer it. You take it in the laboratory so that it can start to grow there. Or you get the egg, you start to grow it like that. You are going to get a sperm from the man, egg from a woman, you put them together, it start to grow. Later on, sometimes you can as well get to put that into the woman back. Or you can use the gamete intrafallopian transfer, where you introduce oocytes and sperms into the ampulla direct. Okay, the oocyte and the sperm direct. There's a medicine that can also be used for those who do not ovulate. Anyone who knows the medicine, we've been talking about it for long now. Anyone who knows the name? So sometimes in males, infertility can be due to ejaculate that contains little sperms, that is known as oligospermia. Or even you have ejaculated, the ejaculate only contains fluid, there is no sperm, that is azoospermia. Okay, and for you to overcome this, can use intracytoplasmic sperm injection. So like you only get sperms and then you are going to be able to inject them in a female like that, because the man cannot release any. Okay, so we reached at the stage of cleavage where the egg has formed, the egg has merged with the male, and then the sperm, they've formed a cell that has got two cells inside. That is known as a two-cell stage. So once the zygote reaches a two-cell stage, it will start dividing. And these cells, which become smaller with each cleavage, are known as blastomeres. So those two cells that you saw, they are called blastomeres. Okay, these cells you can see inside. You can see that the entire cell here is not changing in size. What is just changing in size are the blastomeres, Because they were 2, they will become 4. 4, they will become 8. 8, 16. 16, 32. 32, like that. So until the 8 cell stage, they are going to form a loosely arranged clump. So when you reach the 8, when the cells divide and then they become 8, it will form like a bow, like a bow. This thing is not in 2 dimension. It looks like this. It's like a bow. Okay, 8 cell stage. And then after the third cleavage, blastomeres will maximize their contact with each other, and that will form a compact ball of cells held by tight junction. This process, compaction, segregates the inner cells which communicate extensively by gap junction from the outer cells. So we're going to have the cells which are more inside and the cells which are more outside. And on the third day after fertilization, cells of the compact embryo, those which are inside, again will divide to form 16 cell stage. So remember, we move from 2 to 4 to 8 and then to 16. Now the 16 cell stage is specifically called modular because it looks like mulberry. You know a bunch of mulberries, how they look. So that's why it's called modular 16 cell stage. So the inner cells of the modular will constitute what is known as the inner cell mass. And then the surrounding cells which are out will make up what is known as the outer cell mass. The inner cell mass will be what will give out what we call the embryo proper, where the child is going to come from. The outer cell mass will form what we call the trophoblast, which will give us the placenta. So these cells that you can see like this, got the cells outside, which you call the outer cell mass, the ones which inside, inner cell mass. The inner cell mass will form the embryo proper, outer cell mass, trophoblast. Is there any question? Okay. You need to understand this, alright? You need to understand because questions are interesting the way they're asked. So we have formed the module around the third day, and then we are going to have blastocyst formation. So when you have got the modular, you are going to start having a cavity start to develop inside the modular, and that cavity is going to start having fluid penetrating it through the zona pellucida, which is the cover. Remember, the zona pellucida disappears by the fourth day. Okay, so when the fluid starts to enter into the intercellular space, the space between cells or the inner cell mass, it forms that big cavity, and that big cavity becomes what is known as a blastocele. And when you form the blastocele, the whole embryo is now called the blastocyst. So take note of how names are changing. It was 2 cells, it became 4 cells, it became 8 cells, it became 16, which is the modular, and then it became 32, which will have a blastocele, which is a cavity, and that is known as a blastocyst, also known as a blastula. Blastocyst or blastula. The blastocyst can become even much bigger. The space inside the blastocyst can even become much bigger, it's known as a blastocyst. You can see you have got the cells which are inside, that is known as the inner cell mass, also known as the embryoblast. And then the cells which are outside, which is known as the outer cell mass, which forms the trophoblast. You can see that the cells which are inside will concentrate on only one side of the cell. That's the embryoblast or the inner cell mass. And then the trophoblast or the outer cell mass are surrounding the entire cell. So cells of the inner cell mass are now called the embryoblast, and they're at one pole. And those of the outer cell mass, they are called the trophoblast. They are going to become flat and they'll form an epithelium of the blastocyst or the blastula. The zona pellucida has now disappeared. This should be the fourth day, and that is going to allow implantation to begin. In the human, trophoblastic cells over the embryoblast or begin to penetrate between the epithelial cells of the uterine mucosa about the 60th day. So implantation happens around the 60th day. You can see the cells which are near the embryoblast are the one which are starting to penetrate the wall of the uterus. Okay, the wall of the uterus, and that is how implantation is going to be happening. So sometimes there can be abnormal zygote formation. The exact number of abnormal zygote formed is unknown because they are usually lost within 2 to 3 weeks of fertilization. So if you are an abnormal zygote, you will not even know that you were pregnant because 2 to 3 weeks they would be terminated. Okay? So estimates are that as many as 50% of pregnancies end up in spontaneous abortion without even knowing. And the majority of them are because of chromosomal abnormalities. This is a question which they will normally bring. Majority of abortions that happen automatically are due to chromosomal abnormalities. So these abortions that happen naturally are actually good because they are more like for screening. You don't want what is not capable to implant, because if it implants, you know what will happen? It can lead to congenital malfunction. Someone is born without a mouth, without legs, and everything that can go wrong. So it is a protective mechanism. So as a quick summary, what is happening? There is cleavage that is happening, right? Cleavage is a series of mitotic divisions. There is mitosis happening. The zygote, remember it's mitosis. Mitosis is multiplicative. 2 cells, 4, 8. Okay? And so cleavage in humans is holoblastic, meaning that the cell is going to divide completely through the cytoplasm. And it is also asymmetrical. Daughter cells are unequal in size. One of the cells is going to get a bigger cytoplasm than others. And the cleavage is also asynchronous. Only one cell will divide at a time. Not all the fertilized cells or all the cells would divide. So the largest daughter cell would divide next, at least during the few the next few divisions. Now the process of cleavage will form a blastula which consists of cells which we call the blastomeres, right? Blastula, also known as blastocyst. At 16 cells is known as a morula. Take note that blastomeres are totipotent up to 8 cells stage. What do we mean by totipotent? Each blastomere, each one of those cells which we are seeing inside up to the 8 cell stage, you can actually get one of those cells, it can still form a complete human being. Only one, okay? Because each blastomere can form a complete embryo by itself. That's totipotent. Totipotence refers to a stem cell that can differentiate into every cell within the organism, including extra embryonic tissue, the tissue that supports the embryo. This comes in the exam. So blastocyst formation is going to involve the fluid secreted within the morula that forms the blastocyst cavity. The conceptus, that thing that is going to be born at this time, is known as a blastocyst. It's not yet a baby. Blastocyst. And then the inner cell mass of the blastocyst is called the embryoblast. The embryoblast, the inner cell mass, is pluripotent. Please take note. Blastomeres up to 8 cell stage are totipotent. The embryoblast is pluripotent. What does it mean? It is going to give you 3 germ layers, the ectoderm, the mesoderm, and the endoderm, where all the body organs and everything that you see will come from. The outer cell mass is called the trophoblast. We talked about that. Zona pellucida will degenerate by the 4th day. So this is what you have. You can see the fertilized egg up to the 8 cell stage is totipotent. The embryo blast, the inner cell mass, is pluripotent. And then when these inner cell masses, you form now a baby. Baby is going to have other cells from where other things can come from. For example, hematopoietic stem cells, where all blood cells can come from. Those are multipotent. Okay. Also, like the neural stem cells, mesenchymal cells, you can get other things from them. These are multipotent. So know what is totipotent, know what is polypotent, know what is multipotent. Where does implantation happen? So when you look at the uterus, at the back, posterior, and on top, that is where implantation happens around the 6th to 7th day. All right, normal 6th day. Now, implantation is going to happen in what we call the functional layer of the uterus, because the uterus is going to have, we talked about the endometrium, myometrium, and the perimetrium, right? Now, the endometrium is going to further have different layers. It's going to have the functional layer and the basal layer. Functional layer is where fertilization, implantation is going to happen. Remember we said that it is actually the part of the outer cell mass or the trophoblast near the embryo where implantation is going to start. So the trophoblast at this point will differentiate and form the cytotrophoblast and syncytial trophoblast. Syncytial, think of a syncytium. It looks like one cell, because you are not going to differentiate the cytoplasm. The nuclei are just going to be like a pool in one cell. That's why it's known as syncytial trophoblast. If implantation does not happen because of immune rejection, known as a graft versus host reaction, we talked about this in immunology if you remember, then so if there is actually an immune rejection, then fertilization, there's not going to be any implantation happening. Okay, so you can see this thing has moved all the way from here. Then implantation has come to happen here in the functional layer. You can see that the trophoblast has divided into two: the syncytial trophoblast and the cytotrophoblast. Look at the syncytial trophoblast. You can see you can't differentiate the cytoplasms. You only have nuclei inside. This is what will be used. This going to stretch out like hands, which will penetrate into the uterus for implantation to happen somewhere on the 7th day. Okay, so you can see how the uterus is implantation happening on the 6th day. So you already know the phases that happen to the uterus during the menstrual cycle. And this is going to be like, you have got the follicular or the proliferative phase, the secretory or progestation, pre-gestation phase, because it's before pregnancy, or the menstrual phase. We already talked about all these phases during the menstrual cycle. But in the proliferative phase, which begins after the menstruation has happened, the uterus will begin to grow because of estrogen. The secretory phase will be like 2 to 3 days after ovulation, after the egg is released. Okay? And then you are going to remain with what is called the corpus luteum. From that thing where the egg was released, that what remains is known as the corpus luteum. If fertilization does not occur, the endometrium is going to be shed. So the endometrium has got 3 layers: the compact layer, the spongy layer, the basal layer. So the compact and the spongy layer, these two forms what we call the functional layer. These are the ones which are shed off. The basal layer is not shed off during menstruation. That will happen in the next cycle. Okay, so you can see the, remember we are saying that the whole uterus is going to have 3 layers: endometrium, myometrium, perimetrium. But the endometrium is also having 2 layers. The functional, it has 2 layers. Yes, the functional layer, which is known as the stratum functionalis, and the basal layer, stratum basalis. But the functional layer has got also 2 layers: the compact layer and the spongy layer. compact is superficial, is small on top, and then the spongy is down. Look at the blood that goes to the uterus. I should talk about this. You can see this is the aorta. Remember we started from the heart. The heart has got the ascending aorta, the arc of the aorta, then the descending aorta. It will be in the thoracic, it will be known as the thoracic aorta. It will penetrate the diaphragm and go into the abdomen, it is known as abdominal aorta. And then the abdominal aorta will continue on down here and split into common iliac arteries, which can split also into internal and external iliac arteries. So from the internal iliac artery, that is where we're having what we call vaginal arteries, because they're going to the vagina, and also uterine arteries because they're going to the uterus. But then you also have arteries which are going to the ovary, which we call ovarian arteries. Ovarian arteries don't come from the internal iliac. Instead, they come from the abdominal aorta. You are going to have from the left and also from the right. Look at what happens. The ovarian artery will send branches which will anastomose with the what? The uterine arteries. Anastomose is they meet. Okay, they will meet. And then you can see these arteries will be able to penetrate the perimetrium, myometrium until they reach the endometrium. And when they reach the endometrium, this is the one. You can see this is the uterine artery. It will form what we call the radial artery. The radial artery will start to coil, to become spiral. First it will be straight. It will give actually straight arteries, and then it will form spiral arteries, which are in the basal layer. You need to know in which layer what artery is there. And then it will also continue like that, forming these which are more even coiled arteries, which will go into the functional layer. Okay, to the functional layer. So these are the ones that you can see. These are the ones that you can see. So I've got the coiled arteries. So what happens is that when the fertilization does not happen, I mean if fertilization has happened, the uterus is this big. The egg was released on the 14th day and the implantation has happened. The egg, the embryo has implanted, but from where the egg was removed, that is known as the corpus luteum of pregnancy. The corpus luteum will be the one to be producing progesterone. Progesterone maintains pregnancy. That you need to know. Up to like the 4th month. Okay. So normally the human blastocyst is going to implant in the posterior wall uh, posterior wall of the endometrium. Okay, so what else? If the oocyte is not fertilized, then the venules and the blood vessels are going to be packed with blood cells, and then they are going to undergo extensive diapedesis. You know, diapedesis, this is basically movement of blood into tissue. So when the menstrual phase begins, menstruation, blood is going to escape from the superficial arteries. It will start coming out of the arteries which are just on top there on the endometrium, because when fertilization, if it didn't happen, this layer on top is going to be released. So the arteries on top will be injured, so blood will start coming out. That's why it looks bloodish, okay, during the menstrual cycle. During the following 3 to 4 days, the compact and spongy layer are expelled from the uterus. If fertilization didn't happen, the basal layer, the layer on down, remains. Okay. Thin layer which is supplied by its own arteries, the basal layer, functional layer. The basal layer is going to continue to grow again, and it will again form the functional layer. So the basal layer is never removed. You can see the arteries. This was the ovarian artery. The basal layer, you have got You can see you have got these arteries which are going like that, and then it forms what we call the acute arteries, which join like this. And then from this, we're going to have the radial arteries coming out. Radial arteries coming out. From the radial arteries, we're going to have now basal arteries, you can see, and then spiral arteries, which will go to the endometrium. Spiral arteries. Okay. So as a quick summary, what will happen day by day in menstruation? In day one, on day one, what is basically happening is that fertilization is happening, and you are forming a diploid zygote. That is the one. Day two, what is happening? Cleavage is happening. You're having the cells inside as the blastomeres. Day three, you're having the modular 16 cell stage. Day four, you are forming a cavity which is a blastocele and the shading of the zona pellucida. Day five, you have got the blastocyst. This is the 32 cell stage blastocyst because you have got a big cavity inside there. And then day six, hatching happens. Okay, the blastocyst is going to start coming from its outer shell, which is the zona pellucida. That is what is known as hatching. It is different from shedding off of the zona pellucida. Hatching is coming out of the outer shell. Shedding of the zona pellucida happens on the fourth day, but hatching is happening on the sixth day. Okay, and this is preparing for implantation. The trophoblast cells begin to interact with the uterine lining, specifically the endometrium, not the other linings. And that is known as the apposition. And then the seven implantation really has occurred. The blastocyst adheres to the endometrial lining. Okay, so we've already differentiated hatching and shedding. So basically, that is it. That is about the first week of development. Now, I want you to go and read a bit. In which layer of the uterus does implantation happen? Endometrium, myometrium, or perimetrium? Anyone? Which layer? We have already discussed it. Be free to answer. In which layer of the uterus does implantation happen? It's endometrium, yes. So you go and find out because sometimes the baby can the zygote can pass instead of implanting in the endometrium, it can pass and go all the way to the myometrium into the muscle. Imagine implantation is happening in the muscle. Sometimes it can actually go all the way to the perimetrium. Those conditions have got names. Go and find out what is the name of the condition when implantation happens in the myometrium or in the perimetrium. So you can get look at these questions, okay, from your past papers. We've talked about basically everything, so there shouldn't be a question that you should fail. Where does the blastocyst normally implant? If you fail to answer this one, then the problem is not with us as a team, it's with you as a person. Which of the following is not a glycoprotein forming zona pellucida? I did mention them. Which chronic maternal condition is associated with big babies microsomia? Can someone answer this one? Hypertension, diabetes mellitus, asthma, sickle cell disease. Very good. Now I didn't talk about how you calculate, but I'll talk about this one in the next class. How you calculate? Okay, I can actually just mention it. How can you predict when someone is going to become pregnant? Okay. How can you predict when someone will become pregnant? Like for example, here she tells you that her last normal menstrual period. So if the normal menstrual period was on 26th September 2022, when is this person going to give birth? Likely using the Negele's rule. We'll talk about these rules, of course, but just add 9 months to that period, and then after then, so 26 plus September plus 9 months. Which month is it going to be? September, October, November, December, January, February, March, April, May, June. So it's going to be in June. But to that June 26th, add 7 days. So 26, 27, 28, 29, 30. And then we'd have started July. I've added like 3, 4 days, and then 1, 2, 3. That will be 7 days. So 3rd July, that's when the birth is likely to be happening. So in other words, to the day of your last normal menstrual cycle, the last day you had blood coming out, add 9 months and then add 7 days. Following arteries are only found in the myometrium of the uterus. Yes, please. There's a question? hmm You can go on. Okay. The following arteries are only found in the myometrium of the uterus. So you should be able to answer it. About zonal reaction. hmm Yes. Okay. So after fertilization happens, you are going to have beta human chorionic gonadotropin, beta HCG, that is secreted by syncytial trophoblast, and that will maintain pregnancy. Syncytial trophoblast. Okay. The following is first about the normal menstrual cycle, about the uterine tubes. You can answer that. Longitudinal folds of the uterus mucosa are more prominent in. Following are the actions of estrogen, except the luteinizing hormone surge is important for the following, so look out. Concerning the proliferative phase of the menstrual cycle within the ovary, about the chromosomal reaction, concerning the luteal phase. So try to answer this. The following can be used to predict ovulation except the 21 serum, Spina Beckett, basal body temperature, the 14 estrogen. Which one can you pick? Which one is the exception? Or you have already forgotten? So we are done with the class. I just want to mention one thing within a minute. You need to know the difference between the zonal reaction and the chromosomal reaction. Okay, remember we're talking about acrosomal reaction and zonal reaction. So zonal reaction, acrosomal, starting with where is the acrosomal reaction? Acrosomal reaction allows the binding, okay, of the sperm with the zona pellucida and then it to be pushed in. That's the zonal reaction. Acrosomal reaction is basically the opposite. You want to prevent the penetration of sperms, other sperms, to enter the cell. Zonal reaction allows sperms to enter the oocyte, a sperm actually, not sperms. Then a chromosomal reaction prevents sperm from entering. Just wanted to differentiate that. All right, so that is it for this class. Thank you so much.